Hi there, my name is AureliaReynolds, and I'm going to be presenting the effects of the long protease and the topoisomerase 4 activity in Caulobacter crescentus. So first I want to introduce the Caulobacter crescentus bacteria. It is a gram-negative alpha protobacteria that lives within freshwater environments. The strain that we'll be using for this experiment is the NA1000 or the CV15N. A couple things about Caulobacter is that they have an unusual cell cycle, which we will go further into detail, but this diagram provides a quick insight into the process. Within the Caulobacter, there's a protein called the long protease, which is responsible for degradation and regulation of many different proteins. Through transposon and RNA sequencing techniques, we're allowed to see certain genes in Caulobacter that are essential to the cell's vitality. In a particular knockout called the delta lon, these cells actually lack the production of long protease. And delta lon mutants have some known phenotypes of being increased levels of DNA replication, altered cell cycles, and long filamented cells. Right here, you can able to see the RNA sequencing data and the transposon sequencing data. We'll get into that a little bit further along the line. So right here, we're looking back at the cell cycle. The Caulobacter has two different stages, the stalk cell and the swarmer cell. The swarmer cell actually differentiates into the stalk cell immediately after entering the S phase. This initiates DNA replication and completes the cell cycle process. The swarmer cell then continues to stay dormant within the G1 phase until it differentiates. This is called asymmetric replication. Within each of these phases, we are able to see this, the redox of the cell, where G1 is reduced, S is oxidized, and G2 is reduced. This type of redox regulation actually helps with regulating a lot of different proteins within the cell cycle. Within DNA replication, specifically within the S phase, there are four major regulators: CTRA, GCRA, CCRM, and DNAA. Out of these four, there are two that are affect the replication the most, which are CTRA and DNAA. DNA is responsible for the initiation of DNA replication. Once it's phosphorylated, it's able to bind to the site of replication and open up the double helix in order to recruit the other DNA replication factors. This diagram helps to propose a proposed model, including the recruitment of topo4. So going into topo4, topoisomerase 4 is a protein that is very important to DNA replication. Its main functions are the following: to unwind the supercoils and to decatenate bacterial chromosomes. So basically in DNA replication, as the DNA opens up, it actually creates tension on the side before the DNA replication complex. And this is called the replication fork. And as the replication fork moves, it pulls on the already twisted DNA to begin with. Think about twisting an old telephone coil even further. And it creates these, what we call supercoils, where the DNA actually gets over twisted and provides a lot of tension on the DNA. So in order to stop the DNA from breaking, topo4, topo1, and gyrase go in and help alleviate the supercoils. Another activity of topo4, specifically within bacteria, is it decatenates bacterial chromosomes. So once replication of chromosomes are done, they actually are linked together in a chain-like fashion, almost like this, and topo4 goes and cuts and then rebinds them so that they're able to separate and go into the daughter cells. Topo4 has two subunits, par C and par E. Par C is responsible for the mechanistic activities of topo4, and par E is responsible for the catalytic activity. Topo4 is active in par C and par E are bound. However, par C is able to bind to DNA alone during replication for the relaxation of these supercoils, and par E kind of floats throughout the cell. The decatenation activity happens when par C and par E are bound together at the end of DNA replication. Now, the thing to note is that within this study, par C and par E might be referred to topo4 subunits A and B, respectively. Looking at two other genes that help regulate with topo4 activity, we're going to look at NSTA and GAPR. So looking again at that supercoiling activity, we have topo4 and specifically GAPR. So here we have the relaxation of supercoils. GAPR actually finds these supercoils by binding to a specific type of shape of the DNA and recruiting topo4. Here we're showing the topo4 decatenation activity. Now, NSTA does the exact opposite. It actually binds to the par C subunit and inhibits topo4 activity within this decatenation, and therefore there's no decatenation when NSTA is present. Now, what we're finding is really interesting is within these types of RNA sequencing and transposon sequencing data, we're actually seeing a little bit of a discrepancy. So what we're able to see is that in NSTA, there are no insertions in either the delta lon or the wild type strains. This means that the NSTA is essential for the cell's vitality in both a mutant and a wild type. We're also seeing what looks like a slight upregulation of RNA expression in a delta lon mutant. In GAPR, however, we're able to see that there is slight upregulation, maybe not, but we're able to see these insertions. So with the fact that it lacks insertions in the wild type, but not in the delta lon, means that for some odd reason, GAP-R is disposable in a delta lon mutant, that it's not needed, and the activity of a GAP-R can actually be not used within delta lon. So what are the questions that we're trying to ask? We're trying to ask, how is topo-4 activity related in regulated in Caulobacter crescentus? And furthermore, how is it regulated in a delta lon mutant? If what we're seeing as a result of lacking a lon protease is due downstream effects, as in, does the lon protease directly affect topo-4 activity? And we're also looking for what other genes are important in this topoisomerase 4 regulation. Are the genes that we're seeing, are they upregulated and downregulated in the same fashion as NSTA and GAP-R? And ultimately, how does lon affect the topoisomerase activity in Caulobacter crescentus? I have a couple of hypotheses. The main one is genes that are involved in topo-4 activity will be downregulated in the delta lon mutant, while genes involved with topo-4 inhibition will be upregulated in a delta lon mutant. Some of the phenotypes that are known in delta mutant may result or correlate to this type of topo-4 activity, being longer filamented cells and an abnormal cell cycle. Why are we asking these questions? Well, one answer is antibiotics. Topo-4 is a very common target for a lot of antibiotics. And while Caulobacter crescentus is not a harmful bacteria to humans, studying the topo-4 activity in any fashion hopefully will help to study that topo-4 activity to other bacteria that may be harmful, and therefore help with the study of antibiotics. And what's really important is that antibiotic resistance is very prevalent in today's world, and so hopefully, any type of study towards that might be helpful. So I have a couple of methods and resources. I'm doing a statistical analysis of raw data, just kind of in Excel. I'm also looking at this integrated genome viewer, which is right here, which is actually showing the raw data of the RNA sequencing and the transponder. So here again, you can see those insertions that are able to show that when the gene is interrupted, it doesn't need to be produced in either one of those strains, but here you can see it's this one, CCRM, is actually essential in a delta lon, but not in a wild type. I'm also using RStudio. We have a couple of volcano plots that are able to show the upregulation and downregulation of these genes in relation to lon and their false discovery rates. I'm also using string network. So string network, we're able to put in all the data and show different types of rich pathways, as well as able to see the different types of genes that are related. So right here is PAR-C, that's topo-4 subunit A, and you can see all the different types of proteins on all the different types of genes that are actually related to PAR-C, either found out experimentally or they're known to be in relation to PAR-C. Lastly, I'm doing a literature review. I'm able to, just collect some information about previous experiments done to help me better understand the topo-4 within the Caulobacter itself. So looking at the statistical data, we're able to see the transposon sequencing on the top and the RNA sequencing on the bottom. And what's really important is that this is actually considered statistically significant. So we're seeing a downregulation of the insertions in the NSTA and an upregulation of insertions of the GAP-R. And with a p-value and a false discovery rate of less than 0.05, these are considered statistically significant. However, in the RNA sequencing, we don't have a high enough fold in order to say that there's any difference. And on top of that, we're not able to say that this is a false discovery. These values are too high. They're over 0.05, so they're not accepted. Looking at the subunits, we're able to see subunit B, subunit A, and again, we're able to see this lack of insertions in both the delta lon and the wild type. Here's some more statistical reads. Again, these numbers really can't be of any significance, unfortunately, just because of the p-values and the FDR values. Lastly, we're looking at DNA gyrase, which is another protein that's in relation to topo-4. Again, you're seeing this lack of insertions again, and you see these insertions right here, but in both subunits, for gyrase, we're not seeing any insertions, which is interesting. Again, these values cannot be accepted just because of the p-value and FDR rates, but I think I would include them anyways. Here we're going to show a volcano plot of some of the genes that are upregulated in DNA replication and how they're important. And see, we have some of our target genes that we know up here. So what do we know? We know NSTA and GAP are affected by lon in some way. We just don't know how. We know that there are numerous other genes involved in DNA replication that are also affected by lon. And also in a study by Zenner et al., we also know that lon actually degrades DNA-bound proteins. So that's anything that binds to the DNA at all. So what is proposed at this moment? Topoisomerase 4, PAR-C, and PAR-E are conserved genes. They have to be in a Caulobacter cell in order for the cell to be viable. We also know that this is the same for gyrase subunits A and B. And I think that the insertions from GAPR are due to it being a DNA-bound protein. So there's a lot of future direction with this. Obviously, my results are not complete. So what I want to do is I want to investigate further in the genes related to DNA replication. This is the list I have so far, but I want to look into it further, and I want to see what trends they are. So obviously we have these trends. I just want to see what kind of correlations they're going to be making. There is a lot of time to tell them about that. There's a lot more investigation to be done on this project specifically. My results are not anywhere near complete, but hopefully they will be. I just want to give special thanks to Dr. Chian for providing such a great environment to work in for the past 2 semesters. I want to provide special thanks to Dr. Auerbach and Dr. Furman for their endless support through all 4 years of the ICONS program. Julia Mendel, she's been a wonderful peer for fantastic support and feedback, and obviously my family for always supporting me through my time at UMass. For questions, you can either email me at any time at the email provided below, or I will be holding a question session on April 26th from 6 to 7 p.m. via Zoom. If you want to attend, just please email me and I will send you a link. Thank you.